Team number 14, Mycoplasma and Ureaplasma. Plan of the team: properties of Mycoplasma, epidemiology of non-gonococcal urethritis, mycoplasmosis and ureoplasmosis, pathogenesis of non-gonococcal urethritis, mycoplasmosis and ureoplasmosis, clinical manifestation of mycoplasmosis and ureoplasmosis, and laboratory diagnostic methods of non-gonococcal urethritis, mycoplasmosis and ureoplasmosis. Mycoplasma taxonomy: order Mycoplasmatalis, family Mycoplasmataceae, genus Mycoplasma, species Mycoplasma pneumonia, Mycoplasma hominis, and Mycoplasma genitalium. And the next genus is Ureaplasma, species of Ureaplasma, Ureaplasma ureolyticum. Mycoplasma and ureoplasmas are classified in the class Mollicutis and order Mycoplasmatalis. And the genus of Mycoplasma contains over 110 species. They exist as commensals, parasites, and pathogens of plants and animals, including mammals, insects, birds, and reptiles. 7 Mycoplasma species are commonly found in humans. Mycoplasma salivarium, Mycoplasma oral, and etc. And the genus Ureoplasma contains 5 species. Of this, only one species, Ureoplasma ureolyticum, may be recovered from human genital and respiratory tract. This species comprises 14 serotypes. And next is Mycoplasma morphology. Size: they are very small, 150 to 350 nanometers in size, and they are filterable by bacterial filters. Absence of precursor of peptidoglycan, and they lack a rigid cell wall, which is replaced by a triple-layered cell membrane containing sterol, and pleomorphic. They are highly pleomorphic, exist in coccoid, bacillary, or filamentous, or even in helical forms. They are parallelogram negative, beta stained by Giemsa stain. They reproduce by binary fission and budding, and they are non-sporing and non-flagellated, non-motile. On the right side of this slide, you can see structure of Mycoplasma. Surface of Mycoplasma consists of lipoprotein membrane, and inside of Mycoplasma soluble proteins present inside of Mycoplasma, and ribosomes, DNA, and soluble RNA inside of Mycoplasma. And next is Ureoplasma and Mycoplasma genitalium morphology and general characters. Mycoplasma and ureoplasmas are very small, measuring 0.2 to 0.3 micrometers in diameter. Therefore, they can pass through the bacterial filters. They differ from other bacteria in that they lack a rigid cell wall. These organisms are bound only by a single trilaminar cell membrane that contains a sterol. Mycoplasma ureoplasma cannot synthesize their own cholesterol and require it as a growth factor in the culture medium. Because they lack a rigid cell wall, they are extremely pleomorphic, varying in shape from coccoid to filamentous and other bizarre forms. Also, because of the lack of bacterial cell wall containing peptidoglycan, these organisms are intensive to cell wall active antibiotics such as penicillins and cephalosporins. They possess a small genome, molecular weight 5-10 daltons. Therefore, these organisms have limited biosynthesis, biosynthetic capabilities, and hence for their cultivation media enriched with precursors for nucleic acid, protein, and lipid biosynthesis, and with the exception of Mycoplasma, a sterol component are required, and they multiply by binary fission. However, cytoplasmic division may lag behind genome replication. This results in the formation of multinucleate filaments and other shapes. They don't possess flagellae or pili, but some mycoplasmas, including, for example, Mycoplasma pneumonia, exhibit gliding motility on liquid-covered surfaces, and this is attributed to specialized tip structures that also help the organisms in the attachment to the host cell. They stain purely by gram stain and are gram-negative. They stain well with Giemsa and Dien's stain. And the organisms of the genus Ureoplasma can hydrolyze urea, while other genera cannot do so. The organisms of the genus Anaeroplasma are strict anaerobes, and those of the genera are aerobes and facultative anaerobes. Most species on solid media form characteristic small fried egg colonies. Next. Mycoplasma biochemical reaction, biochemical tests, glucose, arginine, and urea test. And Mycoplasma pneumonia and Mycoplasma fermentis, Mycoplasma genitalium, and Mycoplasma lightlavi, and utilize glucose and other carbohydrates. And the Mycoplasma salivarium, Mycoplasma oral, and Mycoplasma hominis and Mycoplasma fermentis utilize arginine as a major source of energy. Ureoplasma ureolyticum produces urease enzyme that hydrolyzes urea to ammonia, and the liquid culture medium is supplemented with one of these three substrates and a pH indicator, phenolurate. The carbohydrate fermenting species catabolize glucose to produce lactic acid, resulting in a shift to an acidic pH, and arginine metabolizing species produce ammonia, carbon dioxide, and adenosine triphosphate by metabolism of arginine, and production of ammonia results in a shift in pH to alkaline. And similarly, Ureoplasma ureolyticum produces urease enzyme that hydrolyzes urea to ammonia, again resulting in an alkaline pH shift. And the antigenic properties, Mycoplasma pneumonia possess cell membrane-bound glycolipids and proteins which act as heptans. Glycolipids induce antibodies that react in complement fixation test and other in vitro antigen-antibody reactions, and the glycolipids with similar antigenic structure have been found in human brain, and antibodies of Mycoplasma pneumonia glycolipids may cross-react with brain cells leading to cell damage, and this may account for neurological manifestations of Mycoplasma pneumonia infection. And the susceptibility to physical and chemical agents of Mycoplasma. Mycoplasmas are killed by heating at 56 degrees Celsius for 30 minutes. They can be preserved at minus 70 degrees Celsius or in liquid nitrogen. Mycoplasmas are believed to be resistant to ultraviolet light and the photodynamic action of methylene blue, and antiseptic solutions such as chlorhexidine and citramide inhibit the growth of mycoplasmas. And Mycoplasma pneumonia also possesses two major surface proteins, including the PI protein that mediates attachment, called agglutinins that are induced in patients with Mycoplasma pneumonia infection are directed against one antigen determinant of erythrocytes, leading to an autoimmune response and damage to erythrocytes. And culture characteristics of Mycoplasma. You can see in this big slide colonies of Mycoplasma on agar showing fried eggs appearance. Laboratory diagnosis of Mycoplasma and ureoplasma infections may be carried by culture and serological test. And culture technique. Mycoplasma pneumonia may be recovered from throat swabs, nasopharyngeal swabs, sputum, throat washing, and bronchoalveolar lavage, tracheal aspirate, and lung tissue specimens. And next is genital mycoplasmas may be isolated from urethral and vaginal and cervical swabs, and semen, prostatic secretions, urine, blood, cerebrospinal fluid, amniotic fluid, respiratory tract secretions, and synovial fluid, pericardial fluid, and biopsy specimens from endometrium, fallopian tubes, placenta, and aborted fetus. Because of the fastidious nature of these bacteria, the culture media should be inoculated as soon after collection as possible. Standard Mycoplasma broth medium dispensed into small vials may be used for transport of swab specimens, while other specimens may be transported in sterile screw-capped containers. In the laboratory, if inoculation is not possible immediately, then the specimen may be held up to 24 hours at 4 degrees Celsius. If delay more than 24 hours is accepted, then the specimen should be frozen at minus 70 degrees Celsius. Urine specimen should be centrifuged and deposited inoculated into the culture medium. If inoculation is not immediately possible, then urine deposit should be diluted with equal volume of transport medium and frozen. Mycoplasma broth medium containing penicillin, polymyxin B, and amphotericin B, and glucose and phenol red as pH indicator is inoculated with the specimen and incubated at 37 degrees Celsius, preferably in an atmosphere of 95% of nitrogen and 5% of CO2. If specimen contains Mycoplasma pneumonia, its growth is revealed by turbidity and a color change, red to yellow, of phenol red due to fermentation of glucose, and it usually occurs in 28 days. Ureoplasmas and other mycoplasmas which do not ferment glucose will show only turbidity. It is then subcultured on agar medium and incubated in air for 5-7 days. Mycoplasma pneumonia form large 200 to 500 micrometer colonies with a characteristic fried egg appearance, while colonies of ureoplasmas are small, 15 to 60 micrometers in diameter, and usually lack peripheral zone. Colonies of Mycoplasma pneumonia may be identified by hemoadsorption test and tetralism reduction test and serological technique. Epidemiology of non-gonococcal urethritis, mycoplasmosis, and ureoplasmosis. Mycoplasma hominis, Mycoplasma genitalium, and ureoplasma urolyticum are associated with urogenital tract disease. They frequently colonize female lower urogenital tract, such as vagina, periurethral area, and the cervix. transmissions, they are transmitted mostly by sexual contact or mother to fetus during birth. Pathogenesis of non-gonococcal urethritis, mycoplasmosis, and ureoplasmosis. 5 vaginal patterns lead to first exposure to Ureoplasma ureolyticum. Bacteria are able to establish infection and increase in titer. And the next process is inflammatory response resulting in neutrophil influx, accounting for signs of infection and associated symptoms, leads to chronic colonization along with signs and symptoms. This leads to resolution of symptoms, and chronic colonization along with signs and symptoms lead to natural appearance of infection, followed by further sexual partners and subsequent exposure to Ureoplasma ureolyticum lead to non-post protective immunity load increases, lead to inflammatory response resulting in neutrophilic influx, accounting for signs of infection and associated symptoms. And the resolution of symptoms lead to chronic asymptomatic urethral colonization, leading to increase in inflammation and reactive oxygen species, malondialdehyde, and subsequent damage to sperm and possible development of antibodies with cross-reactive anti-human sperm membrane proteins. Lead to Ureoplasma ureolyticum associated infertility, and natural clearance of infection followed by further sexual partners and subsequent exposure of Ureoplasma ureolyticum leads to protective immunity, keeps Ureoplasma numbers low and no development of signs of or symptoms. And next is commensals like association with the host with no negative outcomes. And the next is no development or symptoms of signs or symptoms leads to chronic asymptomatic urethral colonization, leading to increase in inflammation and other substances, and this possible development of antibodies with cross-reactive and anti-human sperm membrane proteins. And Ureoplasma ureolyticum may cause non-chlamydial, non-gonococcal urethritis, epididymitis, vaginitis, and cervicitis. However, Chlamydia trachomatis appears to be responsible for 30-50 cases of non-gonococcal urethritis and cervicitis. And Mycoplasma genitalium has been associated with some cases of non-gonococcal urethritis and pelvic inflammatory disease. Its prevalence is not known, but it may be primarily resident of the gastrointestinal tract that occurs secondarily in the genitourinary or respiratory tract. It is very difficult to recover from culture and may require 2-3 months of incubation. And clinical manifestation of non-gonococcal urethritis, manifestation of urogenital mycoplasmas are as follows: non-gonococcal urethritis and epididymitis, mainly due to ureoplasma and Mycoplasma genitalium, and pyelonephritis caused by Mycoplasma hominis, and urinary and urinary calculi, ureoplasma. Pelvic inflammatory disease mainly due to Mycoplasma hominis. Postpartum and post-abortal infection and non-urogenital infection such as brain abscess, wound infection, and neonatal meningitis. And ureoplasmas may cause chorioamniotitis, prematurity, postpartum endometritis, chronic lung disease of the premature infant, and infection of wounds and soft tissues. Ureoplasmas are the commonest organisms isolated from the CNS, central nervous system, or lower respiratory tract of sick, premature, or newborn infants. Ureoplasmas have also been blamed to cause male and female infertility and low birth weight, but there are conflicting reports. Yes, and the Mycoplasma hominis may cause salpingitis, tuber-ovarian abscess, pelvic abscess, septic abortion, and puerperal infection, post-operative wounds, septic arthritis, and septic thrombophlebitis, and peritonitis, and brain abscess. It may cause PAP similar to that caused by Mycoplasma pneumonia and meningitis in newborn. And the laboratory diagnostic methods of non-gonococcal urethritis, culture method, hemadsorption test, serological test, and PCR. Hemadsorption test: colonies growing on the surface of the agar are flooded with 2 milliliter of the 0.2 to 0.4% suspension of washed guinea pig erythrocytes suspended in Mycoplasma broth media. The plate is then incubated at 35 degrees Celsius for 30 minutes. It is occasionally rotated during this time. It is then washed with 3 milliliters of Mycoplasma broth medium by gently rotating the plate. Wash fluid is removed by aspiration with a pipette, and the colonies examined under 50 to 100 magnification, and the colonies of Mycoplasma pneumonia absorb the erythrocytes to their surface. This is due to binding of the Mycoplasma to sialic acid containing oligosaccharides present on the surface of erythrocytes. However, colonies of Mycoplasma genitalium also share this property. Tetrazolium reduction test. Mycoplasma pneumonia has the ability to reduce the colorless compound triphenyl tetrazolium to red compound formazan. The agar surface bearing the suspected colonies is flooded with a solution of 2p iodophenyl 3-nitrophenyl 5-phenyl tetrazolium chloride and incubated at 35 degrees Celsius for 1 hour. Colonies of Mycoplasma pneumonia appear reddish after an hour and may appear purple to black after 4 hours. Other mycoplasmas are negative. And serological technique, these include inhibition of colony development around discs impregnated with specific antiserum, or the fluorescence of colonies treated with such antiserum labeled with fluorochrome. Serological test, there are of two types: detection of antigen and nucleic acid, and detection of antibody. Detection of antigen and nucleic acid, detection of specific DNA by dot blot hybridization and PCR in respiratory exudate, and detection of RNA nucleotide sequences by probe hybridization in respiratory exudate. And the cold agglutinins we use, we use Streptococcus mg agglutination test, and we use complement fixation test. This is the most widely used serological test for diagnosis of Mycoplasma pneumonia and other species of Mycoplasmas. The antigen used is a glycolipid from the organism that is excreted by chloroform methanol. Recent infection is indicated by fold of Morse rise in antibody titer or a single titer of 64 or more. Complement fixing antibodies appear 10 days after infection with the organisms and reach peak titer after 6 weeks. Such results are obtained in about 80% of cases. And we use enzyme-linked immunosorbent assay. Immunoglobulin G ELISA is most frequently used test. It used to detect specific immunoglobulin M antibodies in the patient serum. This test has sensitivity and specificity of 97 and 99% respectively. Recently, a quantitative rapid signal specimen membrane-based ELISA has been evaluated as a rapid diagnostic method and for demonstration of either immunoglobulin M or immunoglobulin G antibodies.